Next, we have Dr. Matthew Grant from Washington University, St. Louis Children's Hospital, presenting enteric nervous system engraftment after in utero hematopoietic cell transplantation. Dr. Grant.
Thank you for allowing us the opportunity to present our work. We have no disclosures. The enteric nervous system forms as the result of neural crest migration that occurs in a craniocaudal direction. In the murine animal model, this craniocaudal migration is completed by gestational age E14, populating the enteric nervous system with both myenteric as well as submucosal neuronal plexi. Our laboratory utilizes in utero hematopoietic cell transplantation, or IUHCT, to introduce hematopoietic cells early in gestation at a similar time point to that neural crest migration completion. Our laboratory has previously demonstrated and presented multilineage engraftment within the central nervous system. For example, a donor-derived Purkinje neuron within the cerebellum of a treated animal demonstrated here. However, IUHCT's capabilities to engraft within the enteric nervous system are relatively unknown. We hypothesized, based on our previous work within the CNS, that early introduction of stem cells in utero would allow for engraftment within the ENS. In order to investigate this, BALB-C mice underwent time-dated mating for 24 hours, with subsequent pregnant dams undergoing a midline laparotomy. Fetuses were then injected at gestational age E14 with 20 million living donor marrow cells via the vitelline vein. The donor source was an adult B6 GFP pool with the presence of green fluorescent protein acting as a post-injection tracking method. As a proof in principle project, this group only enrolled 3 mice as part of the concept here, with 3 mice harvested terminally at 1 month of age, with the resultant chimerism of those mice ranging from 38 to 94%. Bowel was harvested from pylorus to distal rectum. Saline perfusion of the animals was completed, and those duodenal, ileal, and colonic sections were divided into 2-centimeter segments and fixed in 4% PFA while pinned to a silicone elastomer-coated Petri dish that allowed for the bowel to be splayed out into a planar configuration, and after fixation, we're able to separate the mucosal and muscular layers that facilitated individual analysis as well as free-float staining for HuC-HuD, which is a neuronal marker of neurons in the enteric nervous system, glial fibrillary acidic protein, or GFAP, which is a glial marker within the bowel and CNS, and green fluorescent protein, which is a donor-derived marker. The results of those stains is shown here. The HuCD stain demonstrates a significant neuronal network within the bowel with a GFP signal that is a little more scattered with a rare co-localization, as you can see, maybe a handful of neurons that are co-localized. However, a colonic sample from a second mouse in the treatment group, you can see once again the neuronal network in the ENS with a significant overlapping pattern in the GFP channel with co-localization of several neurons within the ENS of both HuCD as well as GFP. An additional colonic sample from the last mouse in the treatment group show a similar pattern of the neuronal network as well as GFP overlay, but there was also several GFP cells that lay in close proximity to the ENS, raising suspicion that these may be existing in a supporting or a glial role. So stain was completed with GFAP, the glial marker. As you can see, the inherent glial network here on the left, with GFP cells taking up in a more scattered random array with no significant co-localization between the engrafted cells and glial cells. Our conclusions were that there's evidence of significant cellular engraftment within the bowel of treated mice after IV IUHCT, with donor-derived neurons present in the ENS of several treated specimens, as evidenced by co-localization with GFP and HuC-HuD. However, interestingly, there were no donor-derived glial cells present within the analyzed samples of this treatment group. Despite the completion of neural crest migration within the murine ENS by gestational age E14, early introduction of adult bone marrow cells through IVA-CT allowed for neuronal engraftment within the ENS. These findings have tremendous translational potential, and that diseases with known gastrointestinal motility or innervation could be treated with this treatment modality, and donor-derived neurons would exist and be able to cross-correct by expressing enzyme and rescuing those deficient cells around them. I'd like to thank our funding agency as well as the people who helped to complete this project and take any questions.
Thank you, Dr. Grant. We'll open this to questions. So my question is this has awesome, potential for therapeutics, obviously, but how do you envision kind of rolling that out if you were to look, in the future and see how we're going to actually treat patients? Because there is a small, very small subset that you're even going to be able to predict are going to have these prenatally. So how do you envision this working?
So we are able to prenatally diagnose several neurogenetic diseases. We think of them as terminally most significant in their central nervous system effects, but we know that lysosomal storage diseases, as presented here already, have significant effects outside of the central nervous system. These kids that get palliated with bone marrow transplant go on to develop significant GI motility issues as well that we can pick up prenatally. That would be a then targetable area that we could do this treatment in utero and both offset, ameliorate the central nervous effects as well as the ENS effects. So we could treat the most significant effects as well as the kind of lifelong things that hopefully this treatment would allow them to have a durable result.
It has a lot of potential. I'm looking forward to seeing you guys do bigger things with this. Any other questions? Thanks, Dr. Grant.